we're gonna cover the higher level content for enzymes and metabolism. First, what is the difference between intracellular and extracellular reactions?
Intracellular is inside the cell and extracellular is outside the cell.
Perfect.
Is that it?
Next, talk about the generation of heat by reactions of metabolism.
What's that supposed to mean?
Metabolic reactions generate heat as a byproduct. Explain why that's important.
To stay warm? I don't know.
Yeah, that's actually right. This is how mammals and birds have a body temperature higher than that of its surroundings. Contrast linear and cyclical metabolic pathways for me.
Linear goes in a line and cyclical goes in a circle.
Can you be a bit more specific?
It's like in cyclical, whatever you get at the end is used for the first reaction, if that makes sense.
Yeah, the starting molecule is regenerated at the end, whereas in linear, the end product is different from the starting molecule. Can you give me more examples of each?
Examples?
Think respiration, for example.
Oh, Krebs cycle and Calvin cycle. And for linear, we have glycolysis.
Great! Next, contrast competitive and non-competitive inhibition.
Oof, a little hint.
First, give me a definition of an inhibitor.
Something that decreases enzyme activity.
Good! Remember active site versus allosteric site?
Oh, yes. The active site is where the substrate binds to, and the allosteric site is like just somewhere on the enzyme that is not the active site. Oh, competitive inhibition is when the inhibitor binds to the active site, stealing the substrate spot. Non-competitive inhibition is when the inhibitor binds to the allosteric site. It changes the shape of the active site, preventing the substrate from binding.
Perfect! Remember the graphs for competitive and non-competitive inhibition?
I think it was something like this.
That's right. Can you explain why?
I forgot.
Think about why competitive inhibition can reach the maximum rate of reaction.
Oh, because when you have enough substrate, the inhibitor doesn't get a chance to bind to the active site.
Good! And for non-competitive inhibition, the inhibitor can bind to the enzyme even if the substrate is bound to it as well. So it reduces the number of functional enzymes and you're never gonna reach the maximum rate of reaction. We're almost done. Explain feedback inhibition.
Is it the end product inhibition?
Yeah.
It was that the end product inhibits the first enzyme in the metabolic pathway.
Great! And how is that useful?
How was that useful?
If you have too much of the end product,
the metabolic pathway will be inhibited, so less of the end product will be made.
And when there is not a lot of the end product,
the metabolic pathway will continue to produce the end product.
Good! This means that the concentration of the end product is held constant.
Got it.
Last one. What is mechanism-based inhibition?
Good question. What is it?
It's when the inhibitor binds irreversibly to the enzyme, making the enzyme no longer functional.
Oh, okay.
Well done for staying until the end.